The ability to tolerate anoxic conditions is a survival mechanism of many aquatic vertebrates. However, the painted turtle exhibits remarkable tolerance to oxygen deprivation and has been recorded to be able to survive up to 4 months without it. But why? There are two physiological mechanisms the painted turtle has adopted to allow for its survival throughout long periods of submergence.
The first of the two adaptive mechanisms used by painted turtles is their ability to greatly depress their metabolism. As an ectothermic reptile, the painted turtle's metabolism will be lower naturally and will decrease in response to decreasing temperature. Although at temperatures of 3 degrees Celsius, the painted turtle is able to decrease its metabolism by 90% within the first 30 days of hibernation. For comparison, that's nearly 10,000 times lower than a mammal of similar size at euthermic temperatures. The painted turtle will decrease their heart rate, blood pressure, and renal function to decrease their metabolic needs. They will also exhibit little to no motor activity during these months of hibernation. During these months of anoxia, the turtle will not have access to oxygen. As a result, they will need to employ the use of anaerobic glycolysis to produce ATP, which uses glycogen as its substrate and results in the production of potentially harmful lactic acid. Because this process is required to maintain the turtle throughout hibernation, it becomes vitally important that substrate depletion and lactic acid accumulation are decreased. In order to decrease substrate depletion, the painted turtle will decrease their ATP production and ATP utilization, which in turn will decrease the level of lactic acid accumulation. To reduce ATP production, the painted turtle will lower the activity of their glycolytic enzymes such as pyruvate kinase, which is used in the final step of glycolysis. To reduce ATP utilization, they will allow for the downregulation of the activity of transmembrane ion pumps, such as the potassium channel, that requires ATP to function. The overall activity of these mechanisms will allow for the reduced metabolism of the painted turtle throughout its anoxic hibernation.
The second mechanism relies on acid-base reactions within and surrounding the shell. Turtles undergo anaerobic respiration as a way to synthesize ATP for their bodies to use sparingly throughout their long months under the ice. This process consumes glycogen slowly, allowing for the animal to use it throughout its entire period of submergence. However, long periods of time in anoxia causes an accumulation of lactic acid, the end product of anaerobic glycolysis, to be produced within the tissues of the animal. In order to prevent this, the body uses precautionary mechanisms to buffer or decrease the effect of the potent substance, which are greatly adapted to the turtle itself and are not present in any other aquatic vertebrates. The painted turtle has various internal mechanisms that aid in the stabilization of blood pH due to the large amounts of lactic acid within the body. The turtle has two different mechanisms it uses to combat non-respiratory acidosis. Firstly, the turtle has large amounts of bicarbonate, a highly effective buffer, within its extracellular fluid, present in concentrations nearly twice that of other aquatic vertebrates. This concentrated buffering solution works to limit the extremely high acidic load. This neutralization works by causing a decrease in chloride ions and an increase in potassium, calcium, and magnesium present within the fluid. Secondly, the shell and skeleton of the turtle act as a type of lactic acid sink, swallowing the lactic acid to prevent buildup within the tissue. In particular, the shell of the turtle not only acts as a protection and defense mechanism, but it also aids in the buffering capability of the animal due to its high mineral composition, almost entirely composed of calcium, magnesium, sodium, and phosphate. This process begins when calcium carbonate is released from the shell into the extracellular fluid to buffer the fluid surrounding the turtle. Due to the long periods of acidosis, the accumulation of lactate gets too high for one mechanism to sustain, thus causing calcium and magnesium carbonates within the shell to mobilize. This helps to supplement extracellular buffering capabilities in times of high acidosis. After this, the acid is brought into the shell and is sequestered there. This mechanism works to pull lactic acid, accompanied by a proton, into the shell, where it is then neutralized by minerals within the shell such as calcium, and the hydrogen ion is neutralized by the carbonate. This reaction in turn produces carbon dioxide, which is released from the shell via extrapulmonary routes and into surrounding water. This essentially has no net influence on the blood pH of the animal, allowing it to neutralize and expel accumulated lactate in small portions until the period of anoxia finishes and the shell can release it.
Overall, the combination of reduced metabolism and the complex buffering system of the shell allows for the painted turtle to withstand long periods of anoxia during its months of hibernation.